Welcome. It's Mr. W, and I am so happy to be live with you on YouTube talking about my favorite thing, which is biology. And I would love it. I'd absolutely love it if those of you who are watching right now, who have really been so kind to take your time to join me here on YouTube, would just post a comment just telling me who you are and where you're from, if you're taking AP Bio, stuff like that. So just please give me a couple of comments so I can see who it is that I'm going to teach this lesson to about this unbelievably cool stuff, which is cell communications. It's some of the greatest stuff in biology. And again, I'm really happy to be here. I think you might know a little bit about me. I'm in Berkeley, California. I'm a very recently retired AP biology teacher. I'm the creator of the Science Music Videos channel and the website learn-biology.com. And learn-biology.com is a complete AP biology curriculum. And you can basically do all your learning of AP biology right there, but that's probably not your situation. You're probably in a class with a teacher, and that's pretty great because what you can do is you can just learn a little bit more from Learn-Biology, and hopefully I can assist you in doing that. I'll tell you a little bit more about the website tonight, but don't want to go overboard with that right now. Right now, what I want to do is start teaching biology. And in fact, if you could type that as a comment, I'm ready to learn biology, that would also fill me with a lot of joy. So again, my plan for tonight is to teach about cell communication, but the fact is that what I'm much more interested in doing is answering your questions. And we've got my website, we've got the whole internet at our disposal if something comes up that you ask about that I don't have resources for in this slideshow. So anyhow, I'm very, very eager and anxious to start teaching. So let's do it. Let me tell you what tonight's show is going to be about. What we're going to do is, first of all, talk about the general features of cell communication. And then we're going to talk about cell signaling systems using G protein coupled receptors. So here are some of the questions that we'll address in the show tonight. We'll talk about things like, not that, there we go. We'll talk about questions like, what are ligands? And we'll talk about what are the three phases of cell signaling. This diagram, if you can't tell what A, B, and C are, then you're in the right place. And that's going to make this a fantastic hour that we're going to spend together. And how steroid hormones, non-polar hormones, are different from water-soluble hormones. This is a model of water soluble hormones, something that could dissolve in water, whereas this is a model of something that can't. And the big thing that we're going to look at is how do G protein coupled receptor systems work? That's what we'll be spending most of our time. So if that's a good plan, you just might want to say, go, Mr. W, let's hear it, something like that in the chat. And again, if you just let me know who you are and what you're doing and what you're learning about in your AP biology class, that would be fantastic. So again, a little bit of interaction is really what this is all about. You ask questions and I do my best to see if I can answer. Also, it would be fantastic if you gave me some feedback about things like, can you hear me okay? How's the microphone sound? Stuff like that. Because I think I've got everything set up pretty well. But, you know, I could be talking and you might not be hearing anything. And that's kind of going to be an embarrassing situation. So one of the real ways that I can know about that is through you and you telling me. So anyhow, let's go on. We're going to start by talking about cell signaling, the big picture. In the AP Bio curriculum set up by the College Board, this is topics 4.1 to 4.4. That's in their course and exam description. By the way, if you're looking for an outline to study for the AP Bio exam, I have a fabulous one, and I will put it in the description of this. It's probably not there right now, but I'll put it in the description and you can download it. It's a fantastic study outline. I spent a lot of time doing it because the College Board uses a lot of educational jargon. They're an educational institution, and sometimes that's not so useful to teachers and especially students. So cell signaling, the big picture. On the most fundamental level, what are the two ways that cells communicate with one another? And let's see how we can make this work a little bit better. So basically, there are two ways that cells communicate. And cells are constantly communicating. So this is not like a special case of something that happens in biology. This is all the time. Cells are constantly communicating. And that's true if these are single-celled organisms. We'll talk about in a moment example of bacterial cell communication, or single-celled eukaryotic organisms like yeast, for example, whose cellular mechanisms have been studied in depth. In fact, some of the best models of eukaryotic cell signaling have come about working with yeast. Yeast are single-celled fungi. Or, of course, multicellular organisms, which use cell communication to communicate among the cells within the organism, or, wildly enough, in the case of pheromones, use cell communication to signal between organisms. Kind of amazing. Not going to be something that we're going to talk about very much tonight unless somebody asks about it, and then I'll punt. But if you're interested, just ask a question and I can probably point out a place on my website, learn-biology.com, where I do talk about that. How do cells communicate? There are essentially two ways. Cells can communicate directly, and to do that, they have to touch. And if they touch, there's usually going to be some way for a signal to get from one cell to the next. And that's what we see right here. Or on the bottom, we have communication via signals. And those signals are called ligands. We'll take that word apart in a moment. But here you see a cell that produces some kind of chemical messenger. That kind of chemical messenger, in this case, diffuses through the bloodstream, but it can just diffuse through the extracellular fluid between two adjacent cells, so not even go by blood. And then it'll affect a target cell. Now, how will it affect that target cell? That target cell will have to have a receptor for that signal. So those can be hormones, which are long-distance signals that go through the blood, or they can be local regulators that generally move through short distances. What are ligands? Ligands are signaling molecules, and they're represented here by a circle, but this is biology, and we're talking about molecules that have extremely complex shapes. Many ligands are hormones, but by no means all, and they bind with the receptors based on complementary shape, and that binding leads to a cellular response. A great example of cell communication is quorum sensing. And quorum sensing is a system that's used among unicellular organisms, including bacteria, to coordinate their responses. So we see this in biofilm formation in bacteria, and that's what we see over here. And if this seems a little abstract, that could be your teeth. You wake up in the morning and there's sort of like a little film on the surface of your teeth. That's it. It's bacteria that are kind of piling up on one another. Now, what happens? The bacteria are constantly releasing signal molecules. One of them is shown at number 2. And those signal molecules bind to cytoplasmic receptors. Cytoplasmic receptor. That's a receptor that's inside the cell as opposed to on the cell surface. And we're mostly going to talk about surface receptors, though I'll definitely talk a little bit about cytoplasmic receptors. So here's the receptor, here's the ligand. In the case of quorum signaling, what's really important is how that signal is picked up by receptors in other cells. So imagine that each one of these little cells is putting out a signal, and it's capable of receiving that signal. When the signal exceeds a certain intensity, that intensity is called a quorum. A quorum is used in parliamentary procedure to determine if there's enough people so that you can hold a vote. Then what happens is that these cytoplasmic signals, these cytoplasmic receptors, will actually bind with their DNA, and that will activate genes. And in this case, we see the gene product looks like some kind of polysaccharide. And that polysaccharide creates a kind of slimy matrix that enables these bacteria to settle down. Instead of this is actually called the planktonic. You know that plankton are like little organisms that float around in the sea? Well, this enables these planktonic bacteria to actually settle down on a surface. And the more they settle down, the more of these molecules they put out, and the more the biofilm builds up. Now, is that a bad thing? It's bad for you, because that biofilm formation on your teeth can lead to the buildup of plaque. And one message that I want to emphasize is quorum sensing is an important phenomenon. It's based on the intensity of cells, cell signals that are in a specific area. And the other message is brush your teeth. Brush your teeth every morning, because what you can do is get the biofilm off your teeth by doing that. And of course, you don't need me to tell you that, to brush your teeth every morning. Everybody knows to do that, but you should do that. Now, again, I'm not really seeing any comments come in. So just comment in, say something like quorum sensing, or I'm going to brush my teeth, Mr. W, or something like that, just to tell me that we've got communication going on, because that's really what this is all about. Cell signaling involves 3 phases. List them. I've listed them in this diagram. The first phase is reception of a ligand. Here we have a ligand. It's being represented by a square. But again, think about what you know about proteins. Think about a molecule, think about an enzyme. I was going to say think about a molecule like hemoglobin, which might be something that you can envision. But something that's hundreds of amino acids, it's going to curl into some kind of very, very complex shape. And it's going to bind with a receptor that has a complementary shape. Now, the signal is going to be received, and then it's going to be transduced. What does transduce mean? To transduce is to take a signal and to convert it into another form. I often think about it this way. If someone were to come to the door and ring the bell, that's the signal. I go to the door. I see at the door, there's a package for my wife. And I call, Nancy, somebody needs you to sign. That's me transducing the signal from UPS, the FedEx guy, who knows? The FedEx person, excuse me. So that would be signal transduction. And that produces a second messenger. Again, in the scenario that I just described, me calling out to my wife, that would be second messenger signaling. Or in a way, I'm the second messenger. Though it's a little complex, because in that scenario, I was also kind of like the receptor. But my voice was the second messenger activity. And that's going to lead to a response, which would be my wife coming to sign for the package. I hope that worked for you. And often, before I go further, it's not shown here in this diagram, but there's also often amplification of the signal. In other words, transduction makes the excuse me, initial signal, which can often be one molecule. It can just be one ligand that came to bind. But by the time you've passed through these relay molecules, you can have thousands of enzymes that get activated, bringing about a cellular response. And we'll actually see that when we talk about G protein coupled receptors. Again, not seeing anything in the comment stream, and it makes me a little nervous, but I'm just hoping everything's going okay. And if you had a question or a comment, you would be asking me about that. So let's continue with this. The third phase after the signal transduction is a cellular response. And before I mentioned an enzyme that might be activated. That's one thing that we'll talk about today. Another kind of cellular response can be the activation of a gene. So that's yet another thing that can happen. What happens during the reception phase of cell signaling? During reception, a signal molecule, also called a ligand, binds with a receptor molecule embedded in the cell membrane. Here's a very diagrammatic representation of that. And here's one where the receptor is represented in a slightly more realistic way. The ligand is here represented just as a sphere, but again, biology is all about complementary shapes. I recently released a song about that. I released it on Valentine's Day, which was called a biology lover's love song, and it was all about complementarity, and it is just full of inside jokes for AP biology students. And the cool thing is that at this point in your biology education, you'll understand almost all of those jokes. So check it out, you'll enjoy it. Biology lover's love song, all about complementarity. And as I wrote here, the binding is based on complementary shape. What happens during the transduction and response phases of cell signaling? The receptor interacts with membrane proteins to produce a second messenger. And that second messenger and other relay molecules brings the message to the cytoplasm, where there can be activation of enzymes or the nucleus activating genes. So 3 phases of cell communication. And just so that you know, I also released a rap song that explains all of these phases of cell communication. It's on Science Music Videos in the channel. I think I released it sometime in January. Now, everything that we've talked about relates to water-soluble hormones. So something like, if something was like an amino acid, that's water-soluble because it's got a carboxyl group and it's got an amino group. Molecules of sugar, many carbohydrates are also water-soluble, especially the small ones. But what the question is about is, what happens if you have steroid or nonpolar hormones? The steroids that we know about are, like, for example, when we talk about the sex hormones, testosterone or estrogen. Sometimes if you have allergies, you might use a steroid inhaler, a steroid nasal spray. It's also a steroid. And so how do those work? Well, because they're nonpolar, they can do something that the water-soluble, the polar hormones can't. The phospholipid bilayer, you'll remember, is a lipid bilayer. And like dissolves like, and polar things can't dissolve through the lipid bilayer. That's why they have to bind with a receptor that's on the cell surface. A steroid hormone, which is nonpolar, can diffuse right through the lipid bilayer. How will it have an effect? It's not binding with the receptor on the surface. Rather, what it's doing is it's going to enter into the cytoplasm, where it'll bind with a cytoplasmic receptor. That's what this is over here. And I didn't really represent this very well, but what I tried to indicate is how there's a little groove over here, and that shape is complementary to the shape of the steroid. And here you see the steroid fitting in right over here. That forms a receptor hormone complex. And that receptor hormone complex can pass through a nuclear pore that's built into the nuclear membrane, and then act as a transcription factor and activate DNA. And I surely got a little bit ahead of myself, so I'm going to show the text that's here. So here we have steroid hormones. They diffuse through the lipid bilayer. You can't see that because I need to move over to this view over here, this view over here. They diffuse through the lipid bilayer, and They bind with cytoplasmic receptors, they diffuse into the nucleus, they activate genes, and that's what we see in this step over here. So this, you probably learned this already because of the time of the year. This is acting as a transcription factor, and it's enabling these DNA genes to be transcribed into RNA. That's what I meant by activating genes. And those genes will then, that RNA will then leave through a nuclear pore, go into the cytoplasm, where it'll be read by a ribosome and made into a protein. If you think about the changes that your body might have experienced as you pass through puberty, that was all about your testes or your ovaries producing estrogen or testosterone. I said that in reverse order. The testes produce testosterone, the ovaries produce estrogen. And those go everywhere in your body, but in certain cells, they'll activate, they'll be cytoplasmic receptors. And those cytoplasmic receptors will receive that message and then activate certain genes. That's why if you're male, then you'll develop an Adam's apple like that. If you're a female, you develop breasts. The body will change. There'll be more muscle mass in a male in general, and there'll be other changes. A female will start menstruating. All of those things, that's all about these hormones binding with cytoplasmic receptors that change the activity of the cells. And what we find is that these genetic changes caused by steroid hormones tend to be very long lasting, whereas these changes tend to be pretty quick. So water-soluble hormones bind with receptors, activate second messengers, and that's the whole story. So again, love to know if there are any questions or anything like that. And if there are, just please put them into the chat. I'd love to know. And like, even if there was something crazy going on, like let's say that there was no sound whatsoever because my microphone wasn't working. And I don't really think I have to worry about that. But I'm going to check my settings just in case. I'm going to go right here. And I go audio. And yeah, I got sound. Fantastic. So I'm not going to worry about that anymore. So again, would love to hear some comments or like just see some comments. And that would let me know that people are out there learning biology. And that's something that would make me really happy. So now what we're going to do is we're going to go much, much more into depth. We're going to talk about epinephrine and G protein coupled receptor systems. Before we do that, I want to take a minute, and it's really just going to be a minute. So please stay focused, because this is really something that's going to help you so much in your quest. And if you're here, you're definitely on a quest. What's your quest? Your quest is to do well in your biology, particularly your AP biology course. So how can I support you on your quest? Well, through a website that I created for you. It's called learn-biology.com. And learn-biology.com is essentially an interactive online textbook, but so much more, because a textbook gives you information. This actually teaches you. It actually interacts with you in a way that enables you to learn the material. It includes features like interactive diagrams and click on games. What I showed in the previous screen was that it has complete tutorials about every single topic in the AP biology curriculum. Interactive diagrams and click on games where you would, for example, drag these labels onto various spots. You'll get a positive response if you do it the right way. And if you do it the wrong way, then you'll get some feedback about that too. And that's because our belief is that interaction and feedback is what leads to deep, substantial learning. We have interactive multiple choice questions where if you choose the right answer, you'll get praise. And if you choose the wrong answer, you'll get a hint that'll guide you to what the right answer is. And that kind of interaction, again, is going to lead you to really understand the material. It's going to lead you to memorize the content in a relatively painless way. And we have great resources for getting ready for the AP Bio exam. So let's take a look at what some of those are. And what we're looking at now is learn-biology.com. We're on the website. So what do we have? We have free response questions. We've got 135 of those. We've got flashcards, more than 400 of those. Multiple choice questions, more than 450 of those. And AP Bio Review cumulative click-on challenges. And those click-on challenges are just so incredibly fun. I am gonna, let's see. Okay, so let's see what we would learn if we just did cell communication. This is a little bit risky for me to do live because, man, do I really remember all this stuff? But let's see, let's begin to practice getting the questions. Steroid hormone action, an unbound cytoplasmic receptor. It's got to be this over here, yeah? Great. Now click on a steroid hormone outside of the cell. Got it. Fantastic. Now click on a receptor hormone complex. Receptor hormone complex. There we go. So anyhow, I could do this all day because it's so fun, but you can actually click your way to mastery of biology concepts. And that's because all the interaction is right here. So that's our website. Let's see what else I wanted to tell you about that. And what I want to ask you to do is, I want to ask you to please sign up for a free trial today. Now, in biology, one important idea is mutualism, mutualistic relationships. And this is mutualistic. I, of course, benefit, but you'll benefit too. The free trial, what you would do is you'd go to learn-biology.com, and here's the student free trial page. And once you're on here, you can just learn about all the features of the website and then click free trial. You'll read about our offer, and the offer is a great offer. The offer is an offer that's going to get you to learn so much biology. You click start free trial. That would take you here. And basically, I've already signed up, which is why you're not seeing that. But if I were to log out, then you'd see that you have to, you get a free trial, but you have to give us your credit card first. And you can try it out for 2 weeks. And if you like it, it's $25 a month. It's the best deal in biology education. You will learn so much. You'll feel like the smartest kid in the room, though it's not so important to be the smartest kid in the room. What's important is to be competent and to understand biology, which is the greatest subject. It's going to change the way you feel about yourself. It's going to change the way you feel about this incredible universe we live in. So that's it for this little commercial. Let's now learn some more biology. So the context for what we're going to learn about now, which is G protein-coupled receptor systems, is the fight or flight response. And you might have had the experience where, like, you're walking in a parking lot, going to your grocery store, you're in your own thoughts, you're looking at your phone, and you walk by a pickup truck, and all of a sudden, a big dog throws itself. It's inside the car, inside the pickup truck. It throws itself the window, and the owner left the window a little bit open, and the dog's snout just barely gets through the top of the window, and it's growling at you. Your heart is racing, right? Well, that's called the fight or flight response. And basically what happens is that based on sensory input from your brain, signals go out to the body. One of the most important signals that they go out to are the adrenal glands. The adrenal glands sit on top of the kidneys, and those release a hormone that goes by two names. It's called epinephrine or adrenaline. Now, there are other direct nervous responses, but adrenaline or epinephrine enhances those responses. So again, if you've had this experience, just let me know and say, yeah, Mr. W, I have definitely had that experience. That would be fantastic. And what we're going to talk about, because it's a fantastic example of cell signaling, is how epinephrine is involved in the fight or flight response. So epinephrine, also known as adrenaline, it's a polar or water-soluble hormone. And if you just take a look at the hormone over here, this is a carbon ring with all of these double bonds over here. This is something that you should know as an AP biology student, but if you see this skeletal structural formula, every angle vertex has a carbon, and there'd be enough hydrogens to saturate the carbon. So there's hydrogens here that aren't shown either. But here you've got a hydroxyl group, here's another one, and here's an amine or amino group. All of those make this entire molecule water soluble. And to celebrate the water solubility, I'm going to take a little sip. I'm taking a sip from my Science Music Videos mug, which you could have too. And as I just said, the hormone epinephrine or adrenaline, I'm going to just stick with epinephrine from this point on, is used to enhance the fight or flight response. Just to connect this, though, to your response, to your own experience, if you've ever used an EpiPen, EpiPen is all about adrenaline. So epinephrine's effect are widespread, but they're tissue specific. So your adrenal glands release this hormone. Here are the adrenals. They go everywhere. They go into the blood. They go everywhere in the body. But first of all, only certain tissues pick them up because only certain tissues produce receptors for the hormone. And then because of the genes that are expressed in those different tissues, all cells have the same genes, but they express different ones, then the response will be tissue specific. So there'll be bronchial dilation in the lungs. That means the little tubes that go into the lungs, they'll expand. Well, it's part of the fight or flight response. That's going to enable you to get more oxygen to your cells so you can fight or flee. Your liver will respond to epinephrine by converting its stores of glycogen to glucose. Glycogen is a polysaccharide. It's used for sort of medium-term energy storage in the body. Long-term energy storage is fat. Glycogen is sort of medium-term. Your body can create a couple of pounds of glycogen at the most, whereas the amount of fat that you can put on is essentially unlimited. Why produce glucose? Because glucose is fuel, and glucose will enable your muscles to contract, again, for the fight or flight response. Pupil dilation, why? More information coming in so that your nervous system will make better decisions. Increased heart rate, you're pumping your blood faster, it's delivering oxygen and glucose fuel to the cells. Decreased digestion, because now's not a time to digest, now's a time to fight or flee. Through the blood, epinephrine goes everywhere, but only tissues with certain receptors respond. And the response differs based on tissue type. So what's epinephrine's effect on the liver? Here we have glycogen. The molecule actually will have more glucose monomers than this. I just represented it with about a dozen or so. But what will happen is that changes within the cells will get these liver cells to hydrolyze their glycogen, which is a polysaccharide, into glucose. Glucose diffuses into blood, and it provides energy to fight or flee. And again, that's our theme for the rest of this lesson. How does the hormone epinephrine induce liver cells to perform this feat of breaking glycogen, of breaking glycogen into glucose? So the way that this works is that here are liver cells within the liver. Liver's a large organ that's within your abdomen. On the surface of these liver cells, you have receptors. And these receptors are called G protein coupled receptors. Now, before you saw that barking dog in the parking lot, the liver cells were inactive. That's because there was no epinephrine in the system. So the receptor's unbound. The adjacent G protein, which is part of the transduction system that we're about to talk about, is inactive, as are the other proteins in the membrane, which wind up producing the second messenger, which is cyclic AMP. So essentially, at this moment, the system is off. Receptors unbound, nearby membrane-embedded G protein is off, and nearby enzyme called adenylyl cyclase. Say that with me, adenylyl. It's a beautiful word. Adenylyl cyclase is also off, and the second messenger, which we're going to learn about in a moment, it's called cyclic AMP. It's not active, right? Big picture, G protein coupled receptor systems. This is what they look like before the ligand, which is in this case epinephrine, enters the system. What happens during reception? Epinephrine, we saw the structural formula before, but here we're just representing it as a sphere. It binds with the G protein coupled receptors. Now, these things that look like sticks of dynamite are, in fact, alpha helices that are coiling their way. This is a very typical structure for these G protein coupled receptors, 7 alpha helices. And those alpha helices are embedded in the membranes. This is a transmembrane protein. And there's a cytoplasmic, there's an extracellular receptor over here. And again, what is this? This is actually a bunch of amino acids that take a shape that enable them to bind with epinephrine. If you remember enzymes, you might remember allosteric interactions, where binding at one side of a protein causes changes in another side. That was what brought about a phenomenon called allosteric inactivation or allosteric activation, very similar to non-competitive inhibition. So essentially what happens is that the binding of epinephrine over here ripples through this protein. Think of it as a slinky. Think of it as a big spring. Binding over here, ultimately, nanoseconds later, generates a change over here. Another bunch of amino acids, but their shape subtly changes. At this point, the nearby G protein is still dormant. Now, the G protein in its dormant form is bound to a molecule called GDP, guanosine diphosphate. It's a cousin of ADP. And you remember from the work that you did, maybe you did it with me, in Unit 3, that ATP is active, it's the energized form, and ADP is the inactive form. It's the same thing with this molecule. So here's GDP, that's the low energy form. When G protein is bound to GDP, it's dormant, it's inactive, it's asleep. Reception leads the G protein to be able to bind with GTP. So what happens is that the G protein will interact with the receptor. In all of these diagrams, it looks as if the molecules have intention, but if you go back here, just imagine, the G protein, it's in the membrane, it's a fluid mosaic bilayer. It might move to the left a little bit, it might move to the right a little bit. When this changes shape, then the G protein can actually bind with this part of the receptor. So that's what's happening over here. It touches the receptor's cytoplasmic portion and it sticks. That touch induces a change in the G protein, which causes it to discharge the low energy GDP and bind with GTP. So that's what's going on over there. That activates the G protein. So it was formerly dormant, dormir in Espanol. It's now active. It's awake. Now, once the G protein is awake, it continues to move randomly in the phospholipid bilayer, but nearby is this other membrane-embedded enzyme that's called adenylyl cyclase. So the activated G protein drifts in the membrane, and at some point, it binds with adenylyl cyclase. And that adenylyl cyclase now itself becomes activated. And what it does is it becomes an active enzyme. It only becomes an active enzyme when it's in association with GTP. So what does it do? Adenylyl cyclase's substrate is ATP. It's an enzyme. It has a substrate. It takes ATP and it modifies it. It breaks off these two phosphates over here and changes the shape so that at the end of the conversion process, the product is a molecule called cyclic AMP. Cyclic AMP, a couple of things to know about it. Well, it's still got the same nitrogenous base, it's still got the same sugar, but it's only got one phosphate. And that phosphate is doubly linked to the sugar over here. That sugar is ribose, I believe. So that is why it's called cyclic AMP. There's another form of AMP that's a little bit different, but you don't need to know that. Cyclic AMP is the second messenger in G protein-coupled receptor systems. So what have we done? If you look at this diagram on the lower left, what we've done is we've had the ligand bind with the receptor. There have been changes in the membrane that have led to the release of the second messenger. That's what we've got going on. And I'm wondering if there are any questions. If there are, put them in the chat. I would love to see some questions come on through. So let's review what's happened so far. Excuse me. A ligand, epinephrine, was released by the adrenal glands. It went throughout the body. It bound with cells in the liver because it had a receptor that it was capable of binding with. That binding led to activation of the G protein, which had been formerly dormant. It had had an association with GDP. Now it's bound with GTP. And that enables the G protein to bind with adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP. So that's what's happened so far in our story. And again, what's our story? You're walking in a parking lot. You walk by a snarling dog that surprises you. You're experiencing the fight or flight response. In addition to your heart pounding, in addition to your pupils becoming dilated, in addition to your hair standing up on end, glucose is pouring into your bloodstream. If you were wearing a glucose monitor because you had type 1 diabetes or type 2 diabetes, you'd see a spike in the glucose in your blood. Why is there a spike in the glucose in your blood? Well, it's the downstream cellular response. We're not there, but we're getting there. So again, any questions, please ask. Let's go on. We're now going to talk about the next phase that happens. It's a phosphorylation cascade. So now what we're going to do is we're going to get from transduction of the signal, that was production of cyclic AMP, to the cellular response, which is what happens down here. Cyclic AMP, the second messenger, is going to activate a chain of relay molecules. I know this diagram looks terribly complex. Just calmly watch, and I'll walk you through this. And I, again, also want to emphasize that on learn-biology.com, that's where you'll find an interactive tutorial where you can answer questions about this diagram, get feedback about whether you understand it. So these relay molecules are called kinases, also called kinases. I think both of those pronunciations are completely acceptable. They're certainly acceptable to me. And these kinases are involved in what's called a phosphorylation cascade. Phosphorylation means adding a phosphate to something. And the way this works is it's kind of like a chain of dominoes. One kinase activates another kinase, activates another kinase. And I've only represented a chain of dominoes with 3 dominoes, but these can be exceedingly, exceedingly long with many kinases activated. At the end, we have the terminal enzyme that gets activated, also through phosphorylation, and that is what brings about the cellular response. So now we're sort of piecing this all together. right? Reception, transduction, response. Kinases are activated by phosphorylation, adding a phosphate. So it might be a little bit difficult for you to see, but if you look closely, you can see that this is protein kinase 2. It's the second one in the chain. It's inactive on this side because it's unphosphorylated. But when it gets phosphorylated, it becomes activated and it becomes protein kinase 2 with a phosphate group attached. This phosphate group enables protein kinase 2 to become activated. What does it do as an activated protein kinase? It goes ahead and activates protein kinase 3. Now, why does it activate protein kinase 3? Because of complementarity and shape. In other words, these things aren't like standing in line, but this has a shape that enables it to bind with this and then activate it so that it becomes a phosphorylated protein kinase 3. Protein kinase 3, why can it activate the terminal enzyme? Again, because of complementary shape. So the kinases are activated by phosphorylation, and these activated kinases phosphorylate the next kinase in the chain, and that's called a phosphorylation cascade. And if you can imagine these events, epinephrine being released from the adrenal gland, going to the liver, receptors in liver cells picking it up, activating the G protein activating adenylyl cyclase activating cyclic AMP, or creating cyclic AMP by converting ATP into cyclic AMP, and then this setting off a chain of phosphorylations that goes deep into the cytoplasm. A very, very important thing that you need to understand, and I haven't really represented it, it's very difficult to represent it here, is that there's not just one phosphorylation cascade that's being activated. It's multiple phosphorylation cascades. So what we have is what's called signal amplification. And each step after we get to adenylyl cyclase involves multiple activations. So one molecule of epinephrine binds with the G protein coupled receptor, which activates adenylyl cyclase. Adenylyl cyclase is an enzyme. As long as it's bound to the activated G protein, it's going to keep on taking ATP and converting it into cyclic AMP. So I've only represented 3 here, but enzymes might create hundreds or thousands. And it'll do that as long as the G protein keeps on stimulating it. Well, cyclic AMP is then going to activate multiple phosphorylation cascades. And here's 1, 2, 3, but it's probably more than that. So one epinephrine molecule in this diagram, which ridiculously underrepresents what's happening, we have activation of 1, 2, 3, 4, 5, 6, 7, 8, 9 phosphorylation cascades. And each one of those is activating many of the terminal enzymes. So that's called signal amplification. You start with a whisper, but what you wind up with is a gigantic shouting match where all of these enzymes are being activated at the same time. This is a cell, it's cytoplasm. There are thousands of enzymes. They're all kind of waiting there to do this thing. They don't do it until you walk in the parking lot, you get scared by the dog, and then the dog causes your nervous system to respond by activating your adrenal glands, epinephrine. I mean, I can do this over and over again, but this is an interesting test for you. You should be able to do this. Like, if you can tell this story about how adrenaline activates, how epinephrine activates this release of glucose from your liver cells, then you understand this material. And if you can't, you need to work on it. How can you work on it? Use my AP Bio outline for prompts, but I would suggest that you go to learn-biology.com, because that's where you'll really learn the material in a very, very deep, substantial way. One epinephrine activates millions of enzymes. I think I mentioned this in passing, but the terminal enzyme, the last enzyme in the chain, is glycogen phosphorylase. So that's what happens at the end of the pathway. And when we get to the end, we have activation of the terminal enzyme. It also gets phosphorylated, and that activates the cellular response. If we zoom out a little bit, what's happening? Glycogen phosphorylase, which we saw over here, so hold that in mind, glycogen phosphorylase is now being activated through a phosphorylation cascade. And what it can do is it'll go to glycogen and then it'll snip off one glucose and then another glucose and then another glucose, because glycogen is a polymer that's composed of hundreds of glucose monomers. Those are all going to be snipped off, and those are going to be released into the bloodstream. So this is what it's all about. What we've done is we've gone from release of the hormone to activation of a cellular response that's now leading to an adaptation. And let's see if there's anything else to say. Glycogen phosphorylase converts glycogen into glucose. I want you to think for a minute about this in terms of evolution. This is not a human thing. I don't even think this is it's not a mammalian thing. It's probably a vertebrate thing, and it might just be an animal thing. So this process, and in fact, it might be like a eukaryotic thing, because there's probably some way in which like eukaryotes do it. This process has been happening for hundreds of millions of years, and the fact that we do it and dogs do it the same way, and probably do it the same way as lizards do it, because they probably have their own version of the fight or flight response, and fish probably do it too. These are all what we call in evolution homologies. These are deep cellular homologies that come from the fact that all the eukaryotes have common ancestries. I said that in plural, but really what I meant to say is that all the eukaryotes have common ancestry. So, you saw the dog, it scared you. You had a fight or flight response. Glucose is pouring into your bloodstream. That's good if you needed to actually run away from the dog, but you don't need to run away from the dog. So how does this response get turned off? Well, as quickly as it can be turned on, it can be turned off. And that, again, is part of the adaptive nature of this response. So here's epinephrine over here on the top part of the diagram, and it only bound very weakly to the receptor over here. It was bound by hydrogen bonds based on complementary shape. And so after a couple of seconds, that epinephrine is going to drift off. Now, if you had to continue the fight or flight response, another epinephrine would come in, and that's because it would be pouring out of the bloodstream, and it would cause the response to continue. But, if there's no more activation, then what happens is epinephrine leaves the receptor. Well, if epinephrine leaves the receptor, then this reverts back to its shape. And note that what happened with the G protein is kind of temporary too. The G protein was bound to GTP, but that's kind of an unstable state and a lower energy state. I'm punting a little bit here, but this is sort of my understanding of this, is that without further stimulation, the G protein will also revert back to its former form. It'll release a phosphate group, GTP, which converts that GTP to GDP, and now the G protein is asleep. Well, if the G protein is asleep or dormant, it's no longer going to be stimulating adenylyl cyclase. So end of threat leads to the end of epinephrine secretion. Epinephrine diffuses away from the receptor. The G protein converts GTP to GDP and deactivates. Adenylyl cyclase deactivates. And then there's conversion of ATP to cyclic AMP, and that whole process ceases. In the cytoplasm, there's an enzyme that's called a protein phosphatase, and its job is to take these activated protein kinases and deactivate them. So it removes the phosphate from this activated protein kinase, renders it into its inactive form. And then there's another protein phosphatase that does this over here, and yet another. And I don't know if there are specific protein phosphatases that are specific to each kinase. And if I don't know it, in general, you don't need to know it either as an AP biology student. You just need to know that there are these enzymes, protein phosphatases, that deactivate the activated protein kinases and put them back into their deactivated form. So what does that mean? That means that the enzyme can very, very quickly, all these kinases can quickly switch back and forth. And what that means is that within minutes or seconds, glycogen phosphorylase is going to switch back to its inactive form, and it's going to stop chewing up the glycogen that's in your liver cells and releasing glucose. So what will happen? Either you'll metabolize the glucose because you were running and you'll convert it into ATP, or you have other hormones like, for example, glucagon, which responds to a blood sugar spike by encouraging your cells to take up glucose, and then your liver cells and your muscle cells will reconvert the glucose back into glycogen. And that's how the process ends. When these systems malfunction, there can be big problems. And what I'd like you to do is hold on to this thought. There are signaling systems like this that control cell division. And when they malfunction, that can lead to cancer. And holy cow, I'm getting my first comment. Yazan Al-Atiq. Yazan, I am so grateful to you for commenting. I hope you're well. I hope you're having a great experience in AP Biology. And Yazan, if you just got here, I just want to let you know that this lesson is about to end, unfortunately. But heck, anybody got a question? I see that there are a couple people in the stream right now. Put a question in the chat, and I'll answer it. Either it'll be something related to cell communication, or I'll go to the website and answer it. So, yeah, so I am going to teach a lesson about cell cycle regulation. And when cell cycle regulation breaks down, there's a kind of breakdown that's related to G protein receptor systems. It can cause cancer. That ends this lesson about cell signaling. And I want to tell you how grateful I am to you. I want to let you know that on my website, learn-biology.com, you can sign up for a free trial. And if you do, then you can learn a lot. I'm just seeing another. And I'm seeing this really brilliant comment by Nsage. I don't know if I'm saying your name right, but you're a computer scientist. And there is a huge amount of, I would say, analog programming that's going on with all of these systems. And the logic that's involved is probably very, very similar to the kind of computer programs that get written to control computer systems. Though I have to say that puts me a little bit out of my depth, since I am an AP biology teacher, and that's really what I know about. You know, I use computers and I have some familiarity with them. But are there control systems? Absolutely. And in fact, please check my channel. I don't know exactly what day I'm going to do this, but I'm going to pretty soon do a lesson about feedback and homeostasis. And you can see all of the kinds of feedback loops that are involved. And I actually teach them in the context of sort of, for example, how your thermostat controls the temperature in your house. And that's all a programming system loop with positive feedback, negative feedback, so on and so forth. Nsage, maybe you know someone who's taking AP Biology. Please encourage them to go to learn-biology.com and to consider signing up for a free trial. Again, it's the best bargain in AP biology education. You'll learn so much. And learning biology is one of the best experiences that there are. It tells us who we are. It tells us how our bodies work. It tells us what life is really all about. So head on over to learn-biology.com. Sign up for a free trial. Again, it's no risk to you. It's just, you have to give us your credit card, but no risk for you. Try it out for 2 weeks. You'll be astonished at how much you learned. I want to thank all of you so much for coming by, for spending this hour with me. I love teaching biology. I'll do it again. Please go to Science Music Videos and leave a comment. Let me know how this was. Let me know anything that I can do to improve. I'm really grateful to you for joining me tonight, and I'm looking forward to the next one. So check Science Music Videos channel for seeing what the next live show is going to be. And another thing is that I also have an app. I didn't really talk about it. It's called Biomania. Let me show you it. If we go here and go here. So just, go to Biomania. Let me make this a little bit bigger for you. Biomania, Biomania. It's on the App Store. It's on Google Play. If you download that, it's completely free. Sign up for, you have to sign up. You have to register in order to use it. And once you do, you'll get on my mail list. And then I will tell folks, and then you'll get on my email list. And I should have an easier way to get on my email list, and I'll figure that out soon. Anyhow, I want to thank everybody who was here tonight. It was really great being with you. Looking forward to seeing you on learn-biology.com. It's all about making learning biology easier. It's all about generating your success in AP biology. So thank you so much. I'll see you next time. Email me with questions. See you on the channel. Take it easy. Mr. W signing off.